Control of ventilation refers to the physiological mechanisms involved in the control of physiologic ventilation, which refers to the movement of air into and out of the lungs. Ventilation facilitates respiration. Respiration refers to the uptake of oxygen and the removal of carbon dioxide. Under most conditions, the partial pressure of carbon dioxide controls the rate of respiration. The most important function of breathing is gas exchange. Thus the control of respiration is centered primarily on how well this is achieved by the lungs. There are four main centers in the reticular formation and other parts of the brainstem that regulate the respiration rate. Inspiratory center, reticular formation, medulla oblongata. Expiratory center, reticular formation, medulla oblongata. Pneumotaxic center, various nuclei of the pons. Apneustic center, nucleus of the pons. The first two centers are present on the medulla oblongata, whereas the last two centers on the pons region of brain. Involuntary control of respiration. Ventilatory pattern. The pattern of motor stimuli during breathing can be divided into inspiratory and expiratory phases. Inspiration shows a sudden, ramped increase in motive discharge to the inspiratory muscles. Before the end of inspiration, there is a decline in motive discharge. Exhalation is usually silent, except at high minute ventilation rates. The mechanism of generation of the ventilatory pattern is not completely understood, but involves the integration of neural signals by respiratory control centers in the medulla and pons. The nuclei known to be involved are divided into regions known as the following. Medulla ventral respiratory group. The ventral respiratory group controls voluntary forced exhalation and acts to increase the force of inspiration, regulates rhythm of inhalation and exhalation. Dorsal respiratory group. The dorsal respiratory group controls mostly inspiratory movements and their timing. Pons pneumotaxic center, coordinates speed of inhalation and exhalation, sends inhibitory impulses to the inspiratory area involved in fine-tuning of respiration rate. Apneustic center, coordinates speed of inhalation and exhalation, sends stimulatory impulses to the inspiratory area, activates and prolongs inhalate overridden by pneumotaxic control from the apneustic area to end inspiration. There is further integration in the anterior horn cells of the spinal cord. Control of ventilatory pattern. Ventilation is normally controlled by the autonomic nervous system, with only limited voluntary override. An exception to this is Ondine's curse, where autonomic control is lost. However, the information transported from the brainstem to the intercostal muscles and the diaphragm involves the somatic nervous system, as those are composed of skeletal muscle. Determinants of ventilatory rate. Ventilatory rate is tightly controlled and determined primarily by blood levels of carbon dioxide as determined by metabolic rate. Blood levels of oxygen become important in hypoxia. These levels are sensed by Cham receptors in the medulla oblongata for pH, and the carotid and aortic bodies for oxygen and carbon dioxide. Afferent neurons from the carotid bodies and aortic bodies are via the glossopharyngeal nerve and the vagus nerve, respectively. Levels of CO2 rise in the blood when the metabolic use of O2 is increased beyond the capacity of the lungs to expel CO2. CO2 is stored largely in the blood as bicarbonate ions, by conversion first to carbonic acid, by the enzyme carbonic anhydrase, and then by dissociation of this acid to H+ and HCO3-. Buildup of CO2 therefore causes an equivalent buildup of a dissociated hydrogen ion, which by definition, decreases the pH of the blood. During moderate exercise, ventilation increases in proportion to metabolic production of carbon dioxide. During strenuous exercise, ventilation increases more than needed to compensate for carbon dioxide production. Increased glycolysis facilitates release of protons from ATP and metabolites lower pH and thus increase breathing. Mechanical stimulation of the lungs can trigger certain reflexes as discovered in animal studies. In humans, these seem to be more important in neonates and ventilated patients, but of little relevance in health. The tone of respiratory muscle is believed to be modulated by muscle spindles via a reflex arc involving the spinal cord. Drugs can greatly influence the control of respiration. Opioids and anesthetic drugs tend to depress ventilation, especially with regards to carbon dioxide response. Stimulants such as amphetamines can cause hyperventilation. Pregnancy tends to increase ventilation. This is due to increased progesterone levels and results in enhanced gas exchange in the placenta. Ventilation is temporarily modified by voluntary acts and complex reflexes such as sneezing, straining, burping, coughing and vomiting. Feedback control receptors play important roles in the regulation of respiration. Central and peripheral Cham receptors, and Mechanoreceptors. Central Cham receptors of the central nervous system, located on the ventrolateral medullary surface, are sensitive to the pH of their environment. Peripheral Cham receptors act most importantly to detect variation of the oxygen in the arterial blood. In addition to detecting arterial carbon dioxide and pH, Mechanoreceptors are located in the airways and parenchyma, and are responsible for a variety of reflex responses. These include the Herring-Brewer reflex that terminates inspiration to prevent overinflation of the lungs, and the reflex responses of coughing, airway constriction, and hyperventilation. The upper airway receptors are responsible for reflex responses such as sneezing, coughing, closure of glottis, and hiccups. The spinal cord reflex responses include the activation of additional respiratory muscles as compensation, gasping response, hypoventilation, and an increase in breathing frequency and volume. The nasopulmonary and nasothoracic reflexes regulate the mechanism of breathing through deepening the inhale, triggered by the flow of the air, the pressure of the air in the nose, and the quality of the air. Impulses from the nasal mucosa are transmitted by the trigeminal nerve to the breathing centers in the brain stem, and the generated response is transmitted to the bronchi, the intercostal muscles and the diaphragm. Voluntary control of respiration. In addition to involuntary control of respiration by respiratory neuronal networks in the brain stem, respiration can be affected by higher brain conditions such as emotional state, via input from the limbic system, or temperature, via the hypothalamus, or free will. Voluntary or conscious control of respiration is provided via the cerebral cortex, although Cham receptor reflex is capable of overriding it. While breathing can obviously be controlled both consciously and unconsciously, other basic functions provided by the brain stem cannot be controlled voluntarily. Only conscious control of respiratory neuronal networks in the reticular formation can affect other basic functions regulated by the brain stem. Because of the intermeshed character of the reticular formation, e.g., the heart rate in yoga and meditation,